Okay, hello to all of you. Let's discuss chapter number 8. Okay, okay, we're going to slideshow mode. So basically with chapter number 8, we are going to start a new unit. This is a unit on metabolism basically. And in this unit, first of all, in chapter number 8, we are going to discuss some of the basic terms and some of the basic terminology, some of the basic concepts, and then we will proceed to chapter number 9, which is on cellular respiration, and chapter number 10, which is on photosynthesis. So some of the things which we are going to learn in this chapter will make the foundation for chapter number 9 and chapter number 10. So basically, as we have been talking about cells, like the cells are like a factories in which a lot of chemical reactions are going on. And in these chemical reactions, the energy is being produced. And it's the energy which is basically making the life possible. So basically, all the reactions which are going on inside the cells, we call it metabolism. And metabolic reactions, basically, they involve many steps. They are not just like a reaction like from A to B. So they are multi-step reactions. A gets converted to B, B gets converted to C, C gets converted to D. And each reaction is catalyzed by an enzyme. So we will be, in this chapter, we are going to talk about enzymes. We are going to talk about metabolic reactions. So under the metabolic reactions, there are two kinds of reactions. They are called catabolic reactions and they're called anabolic reactions. Catabolic reactions or catabolism means breaking down something. So when the larger molecules, larger biomolecules, they get converted into smaller subunit molecules, those are called catabolic reactions. For example, like if in our food we have eaten proteins, inside our cells, the proteins get converted into amino acids. So if you remember from chapter number 5, amino acids are the subunits of, they are the monomers which make the proteins. So in the catabolic reactions, the large proteins, they get converted into amino acids. And in this process, energy is released. So catabolic reactions, they are breaking up reactions in which larger biomolecules, they get converted into their monomers or their subunits. And in this process, energy is released. On the other hand, anabolic reactions means building up reactions. So anabolic reactions, building up reactions in which these monomers like amino acids, they can link together. The amino acids can link together and they can form proteins. So those are called anabolic reactions. They are building up reactions. So they are the reactions in which the subunits or the monomers, they link together and they form larger biomolecules. They're called anabolic reactions. And anabolic reactions are the reactions which need energy. Okay, so they need the input of energy. And the term bioenergetics is the study of how energy flows through the living organisms. Okay, so we just talked about what metabolism is, and under the metabolism, what are anabolic reactions and what are catabolic reactions. And now let's talk about what is energy. So energy is the capacity to cause some change. And there are different kinds of energies. There's a kinetic energy, thermal energy, potential energy, chemical energy. Kinetic energy is the energy which is associated with motion. Thermal energy is the energy associated with the random movement of atoms or molecules. Potential energy is the energy which is stored in a molecule, in a compound, because of its structure or because of its location. Now, chemical energy is the potential energy available for release in a chemical reaction. So, very important thing to know about energy is, as I said, energy exists in different forms. The energy keeps on changing its form. Okay, the total energy always stays the same. We're going to talk about laws of thermodynamics, in which we talk about the law of conservation of energy. And this energy keeps on changing its form. So you can see here, this is a person who is standing here. Now, because this person is standing at an elevated position, so there is energy which is stored in this person in the form of potential energy. And when this person is going to dive, this potential energy is going to be converted into kinetic energy. And when this person reaches here, again, the energy is going to be stored in this person in the form of potential energy, but definitely the potential energy is going to be less as compared to when this person was at this position. And then this person, when it starts climbing these stairs up again, it's converting the potential energy again into the kinetic energy. And eventually that kinetic energy will be stored when this person reaches here. This kinetic energy is going to be stored in the form of potential energy. So it's all transformation of energy. Thermodynamics is the study of energy transformations, and there are two laws of thermodynamics we are going to talk about. Before that, let's talk about there are two terms we need to know. One is a isolated system, another is a open system. So open system is that system in which energy and matter can be transferred between system and its surroundings. Open system is a system in which there is an exchange of energy and matter between the system and its surroundings. And closed system is that system in which there is no exchange of energy and matter with the surroundings. Now, organisms, all the cells we can talk about, they are the open systems. Okay, so there are two laws of thermodynamics. The first law is the law of conservation of energy. According to this law, the total amount of energy always remains the same, but energy keeps on changing its form. Okay, energy can be transferred, transformed, but it cannot be created or destroyed. So the total amount of energy always stays the same. That's the principle of conservation of energy. So this is the first law of thermodynamics. This is the second law of thermodynamics. So according to the second law of thermodynamics, whenever there is a transformation of energy, when there is a change in the form of energy, there is always an increase in the entropy of the universe. Okay, and what is entropy? Entropy is the measure of molecular disorder or randomness. Okay, according to second law of thermodynamics, every energy transfer or transformation increases the entropy of the universe. Now what are okay, one important thing we need to know is there are two kinds of processes. One are called, and we will talk about what are spontaneous processes and what are non-spontaneous processes, and then we will come back to the concept of second law of thermodynamics again. So spontaneous processes are those processes which can take place on their own, which do not need the input of energy, and non-spontaneous processes are those processes which need the input of energy, which cannot take place on their own. And processes that decrease the entropy are non-spontaneous, and the processes that increase the entropy of the universe, they are spontaneous. For a process to occur spontaneously, it must increase the entropy of the universe, and the processes which decrease the entropy of the universe, they are non-spontaneous. Now here is something important, like if you look at the biological system, so laws of thermodynamics are the laws of, basically laws of physics. So if you look at the biological system, this is one of the very important characteristic of the biological system, is that the living organisms, they are ordered, right? So you can see here one example here, this is one organism, this is another like a cactus. You can see the order, these organisms, they are ordered. We can find the order at every level. Okay. Now the question is, in the biological system, if we see the organisms, they are ordered, they are more structured. So like you know that like amino acids, they get converted into proteins, right, in the anabolic reactions. Simple sugars, they get converted into polysaccharides. So basically in the biological system, one of the character of living organisms is they're ordered. So when we are talking about the organisms being ordered, so being ordered means less entropy. Entropy is the measure of randomness. Entropy is the measure of randomness. So which means that in the biological system, when the suppose amino acids are getting converted into proteins, which means amino acids are, when they're getting converted into proteins, they're getting more structured, they're getting more ordered, and the entropy is being decreased. So does that mean that, because according to the second law of thermodynamics, with every change, with every energy transformation, there is increase in entropy. But in the biological system, what is happening is, with the changes, with the in the reactions, in the anabolic reactions, the macromolecules are becoming more ordered, which means there's a decrease in entropy, right? So does this mean that the second law of thermodynamics doesn't apply to the biological system? Okay, so that's a question. So organisms create ordered structures from less organized forms of energy and matter. Organisms also replace ordered forms of matter and energy in the surrounding with less ordered forms. So basically the question is, does the biological system violate the second law of thermodynamics? So it seems that if we look at a system level, it looks like biological system does not follow the second law of thermodynamics, because with the energy transformations, or with the chemical reactions taking place, the system is going towards more order. More order means less entropy. So at a system level, if we look at a system level, it looks like biological system is not following the second law of thermodynamics, is not following. But if we look at a universe level, it's all written here in this slide. Basically, there are not only anabolic reactions going on, there are many catabolic reactions are also going on. And all those reactions, they are releasing heat. So there is a lot of heat being released in the surrounding, and that is increasing the chaos, the disorder in the universe, and the overall entropy is being increased. Okay, the overall entropy at the universe level, is increased. So it means that the biological world is not violating the second law of thermodynamics if we look at the universe level. So it's very important to understand a system level and a universe level. At the universe level, the biological world is not violating the second law. So entropy may decrease in a particular system, such an organism, as long as the total entropy of the system and surrounding increases. The evolution of more complex organisms does not violate the second law of thermodynamics. That's all what I said. Okay, now we need to know few more terms, and because these things will be required in the upcoming chapters. Okay, so we need to know what is free energy, what is delta G value, then what are endergonic reactions, what are exergonic reactions, what is ATP, what are enzymes. So we need to know all these things, and because only then we can understand the next chapters, which is on respiration and photosynthesis. Okay, so if the biologists want to know if a reaction is going to be spontaneous or it's going to be non-spontaneous. So if a biologist wants to know if a reaction is going to be spontaneous, means if this reaction can take place on its own, or if this reaction needs the input of energy. So how to do that? For that, we need to know the value of free energy. Now, what is free energy? A living system's free energy is the energy that can do work. So it's a living system's free energy is energy that can do work when temperature and pressure are uniform. So and the change in free energy is delta G. Okay, and how delta G is calculated? It's not written in the PowerPoint, but I can explain you. Delta G is the difference of the free energy value of products from the reactants. So delta G can be calculated by subtracting the G value of reactants from the G value of products. That is delta G, and it can be calculated by this equation. Delta G is equal to delta H minus T multiplied by delta S. So H is the change in enthalpy, or the enthalpy is also called change in total energy. T is the temperature, and S is the entropy. So delta S is the change in entropy. Delta H is the change in the total enthalpy or total energy. So if you know these values, and if you plug in the values here, you can calculate delta G. If the delta G value is negative, then it means that the reaction is going to be spontaneous. If the delta G value is positive, the reaction is going to be non-spontaneous. So to understand why delta G negative means it's a spontaneous reaction, because as I said, delta G is equal to G value of products minus the G value of reactants. Okay, so in a reaction, when the reactants react and they form products, delta G, so the reactants are going to have some of their own free energy, products are going to have their own free energy. So delta G value is the difference of the free energy of reactants from the free energy of the products. So if the delta G value is negative, which means that delta G, if it is negative, it means that the reactants, they have more energy in them. So that is why if the reactants have more energy in them, then the delta G value is going to be negative. So if the reactants have more energy in them, which means that they have more energy, they do not need the input of energy, the reaction can take place on its own. So that is why delta G is negative. But if delta G is positive, which means that in this case, the products which are going to be formed, they will have more free energy as compared to reactants. So reactants have to react and form products. If products have more energy, which means that reactants need the input of energy, only then they can form the product. So that is why delta G is positive, and those kind of reactions are endergonic reactions. And endergonic reactions are non-spontaneous reactions because they need the input of energy, and exergonic reactions are spontaneous reactions because they do not need the input of energy. So also important thing to know is when the system has more free energy, it is less stable and it has greater work capacity. Now a person standing here has more potential energy, so it has more free energy, but it is less stable and it has a greater work capacity. So when it comes down, its free energy decreases, it becomes more stable, and now it has less work capacity. So this is an example of a dye molecule. Like if you take a glass of water and put a dye molecule, so you can see in that drop, these dye molecules are all concentrated, so they have high free energy, but they're less stable. So they will all diffuse out so that the free energy decreases and they become more stable. And this is a glucose molecule. It has, because of its structure, it has more free energy and it is less stable, and it gets converted into carbon dioxide and water. In this state, they are more stable but less free energy. As I said, there are two kinds of reactions, exergonic reactions and endergonic reactions. So exergonic reactions are those reactions, exergonic reactions are the reactions in which there is a release of energy. Endergonic reactions are the reactions in which there is an input of energy. Exergonic reactions, they are spontaneous reactions, and endergonic reactions, they are non-spontaneous reactions. So this is an example of exergonic reactions. You can see here the products, they have high free energy. These reactants, they have high free energy. They have more energy in them, and they are going to form the products. And you can see here, these products, they have less free energy. Okay, so when reactants have more energy and they have to form the products with less free energy, there will be release of the energy. So these kind of reactions are exergonic reactions. And here is the example of endergonic reactions. So these are the reactants. They're going to react and form the products. And this is the energy level of reactants and the products. So you can see here, this is the free energy of the reactants and this is the free energy of products. So it means that this much input of energy is required, only then these reactants can form the products. So these reactants need the input of energy. So these kind of reactions are called non-spontaneous reactions because they need the input of energy. Reactions in a closed system eventually reach a equilibrium, and once they reach to the equilibrium, then they can do no work. So you can see here, like this is an basically example, like if you have this kind of a system, here there is more water, here is less water, the water will move from here to here, and once they reach to the same level, this is called equilibrium. At this level, no work can be done and the system has reached to equilibrium. In the open systems, like in our cells, the system is not in equilibrium because there is always an exchange of energy going on. So you can see our cells are like our open system. Cells are our open system. So in this, there is always input of energy and there is always output of energy. So input, output. So our system is like that and it never reaches equilibrium. Okay, the next concept we need to know is ATP. It's a very important concept because these concepts, so whatever I just talked about, the concept of delta G, the change in the free energy, exergonic reactions, endergonic reactions, all these concepts will be coming in chapter number 9 and 10. So it's very important to understand this. Now the next concept I'm going to talk about is ATP, which is adenosine triphosphate, which is the energy currency. So basically ATP, adenosine triphosphate, is a chemical, and in our cells the energy is stored in the form of ATP, and ATP is adenosine triphosphate, and there are 3 phosphate groups which are linked, and it gets converted into ADP, adenosine diphosphate, and energy is released. So we will talk about that. So ATP is energy currency, and in our cells there is a lot of work going on. There's a chemical work, there's a transport work, there's a mechanical work going on in our cells, for which ATP is required. So, for chemical work is basically pushing endergonic reactions. So in our cells, there are a lot of anabolic reactions going on, which need energy. So they're all endergonic reactions. Transport, many things are moving in and out. We have talked in chapter number 7 about the active transport that needs ATP. Then mechanical work, contraction of muscles. And to do work, basically, so in our cells, basically, the reactions which need ATP, so the reactions which need energy, they're called endergonic reactions. So the endergonic reactions are coupled to exergonic reactions. So exergonic reactions are those reactions which release energy. Endergonic reactions need energy. Okay, so those reactions, they are coupled, and this is called energy coupling. So the exergonic reactions and the endergonic reactions, they are coupled in the cell, and they're called coupled reactions. So one reaction releases energy, another reaction takes that energy. So the main energy form is ATP, adenosine triphosphate, and ATP is composed of ribose, which is a sugar. It's a pentose sugar, and it has adenine, and it has 3 phosphate groups. So it's very important to understand the structure of ATP. Very, very important thing to know about ATP is the sugar which is present in ATP is ribose. It is the same sugar which is present in RNA. Okay, so it has a oxygen here. In the DNA, there is no oxygen here. DNA is deoxyribonucleic acid. So this is a ribose sugar, and here on the carbon number 1 is attached adenine group, and here on carbon number 5 are attached 3 phosphate groups. Okay, so phosphate groups, if you remember from your previous lectures, they have negative charge on them. To keep these phosphate groups together, a lot of energy is required. So the energy is actually stored in the bonds. It is actually stored in the bonds which are formed between these phosphate groups. So when this adenosine triphosphate gets converted into adenosine diphosphate, one of the bond is going to be broken. So the bond between these 2 phosphates will be broken, and the energy is released because the energy is in that bond. So ATP, adenosine triphosphate, gets converted into adenosine diphosphate. One inorganic phosphate will be released, plus energy will be released. So this is the way ATP acts as a energy currency in our cells. So basically, hydrolysis of ATP, so the breakdown of ATP to ADP. So you can see here, this is ATP and water is being added, and one of the bond between 2 phosphates will be broken, and ATP will be converted into ADP. Okay, and phosphate will be released, energy will be released. So this reaction is called hydrolysis of ATP. This reaction is called hydrolysis of ATP. Hydrolysis of ATP is a exergonic reaction. In this reaction, energy is released. And there are many reactions in our cells which are coupled with the hydrolysis of ATP, because hydrolysis of ATP is a exergonic reaction. And let me explain you from here. This is an example of a coupled reaction. So here is one reaction in which glutamic acid combines with ammonia and forms glutamine. This is an endergonic reaction. So you can see delta G is positive. So delta G is positive 3.4. Okay. So in this reaction, energy is required. So this reaction is coupled with the hydrolysis of ATP, because hydrolysis of ATP is a exergonic reaction. So glutamic acid plus ATP. Okay. First of all, this glutamic acid will get the phosphate group and it becomes a phosphorylated intermediate. Okay. So this ATP will be hydrolyzed. It will form ADP and phosphate, the inorganic phosphate, and this inorganic phosphate is going to go and bind to this glutamic acid and it is going to form phosphorylated intermediate. And in the next step, this ammonia is going to react, and this glutamic acid here, the phosphorylated intermediate, it's going to be converted into glutamine, and ADP is already formed when ATP is broken, and inorganic phosphate is also released. So this is the way how the reactions take place. And in this reaction, this is an example of a coupled reaction in which glutamic acid plus ammonia, they get converted into glutamine, and the delta G is positive 3.4, and the hydrolysis of ATP, the delta G is minus 7.3. So it's an exergonic reaction. So the coupled reactions delta G will be minus 3.9. So the coupled reaction is a spontaneous reaction because the delta G is negative. The delta G of the coupled reaction is negative. So we have already seen that how the ATP, if you remember from the sodium potassium pump, we have already talked about how the hydrolysis of ATP helps in the pumping of sodium ions from inside to outside and from potassium ions from outside to inside against the gradient, and that is all with the everything happens with the phosphorylation and the dephosphorylation of the protein. And from where that phosphate group is coming, that's coming from ATP. So ATP is required for transport. ATP is required for, like, a lot of vesicles move inside our cells. So we have already discussed about the endomembrane system, and we have seen how the vesicles, they are formed from the ER and how they move to the Golgi apparatus. So these vesicles, they have to actually move from ER to the Golgi apparatus. So how do they move? They move with the help of these motor proteins, and these motor proteins, they basically function with the help of ATP. Okay. So basically ATP is required inside our cells at various levels. There are many functions happening inside the cell: transport, vesicle movement, all these needs ATP. Okay, as I said, there is a cycle going on in our cells. It's called ATP-ADP cycle. So when the energy is required in our cells, ATP is going to be hydrolyzed and it is going to be converted into ADP. Okay, energy will be released and that energy can be used for many things, and ADP is going to be formed. Okay, ATP gets hydrolyzed and ADP is formed, and then this ADP, when we eat food and the energy is released from the food we have eaten, when these proteins, carbohydrates we have eaten our food, they are broken down, they are converted into their simpler components, into the monomers. At that time, the energy which is released, that energy is used, that energy which is released is used to convert ADP back to ATP. So there's a ATP-ADP cycle going on. So when energy is required, ATP will be converted into ADP. When our cells get the input of energy by when we are eating food, then ADP will be converted back to ATP. Okay, the last concept we are going to talk about in this chapter is enzymes, and it's a very, very important concept, very, very important to understand what are enzymes, how do they function, and various factors which affect, and we have also done a lab on the enzymes. So and these enzymes, you will see they are all catalyzing the all the metabolic reactions, which we'll be talking about in the next two chapters. So basically, what is the enzyme? Enzymes are catalysts which fastens the rate of chemical reactions. They make the reactions fast. Okay, a reaction may take millions of years to take place, or may not take place. But if there is a presence of the enzyme, the reaction can take place very quickly. That's the function of the enzyme. So enzyme is a catalytic protein, and majority of the enzymes, they are protein in nature. RNA can also act as an enzyme. So very important concept to know. Majority of the enzymes are protein in nature. Okay. So you can see here, this is sucrose. This sucrose can break down into glucose and fructose only if there is an enzyme called sucrase. Okay, so basically, how does these enzymes function? So how do they speed up the rate of reaction? So basically, for any reaction to take place, there is an input of energy required, and that energy is called activation energy. The function of enzyme is to lower that. The function of the enzyme is to lower the activation energy. Okay, the function of the enzyme is to lower the requirement of the activation energy. So if the reaction is taking place in the absence of enzyme, the reactants needs more energy, and because they have to reach to a transition state. We can explain from here. So these are the reactants, and they have to form these products. So these are the reactants, they have to form these products. So for these reactants to form the products, they have to actually reach to a state which is called transition state. And in this state, their own bonds will be broken, and then the new bonds will be formed. So to reach to this transition state, these reactants, they need input of energy. And that energy is called activation energy. And the function of enzyme is to lower the activation energy. So this is, you can see, this is a reaction which is going on in the absence of enzyme, and this is a reaction which is going on in the presence of enzyme. So in the absence of the enzyme, the reactants need this much energy, and in the presence of the enzyme, they need only this much of energy. Another important thing to know about enzymatic reactions is, first of all, they lower the activation energy. They do not do anything to the value of delta G. Now, what is delta G? So if these are the reactants and these are the products, this much energy is the free energy of the reactants, and this much energy is the free energy of the products. So delta G is the free energy of the reactants subtracted from the free energy of the products, right? So you can see here, when there was no enzyme, the reactants have free energy of this much and the products have free energy of this much. When the reaction is catalyzed with the enzyme, the reactants have free energy this much and the products have energy this much. So enzymes do not do anything to the free energy. The delta G stays unaffected. Okay, some of the terms, basically, how do enzymes function? So the enzymes, they basically act on substrates. Okay, so the reactant that an enzyme acts on is called the substrate, and the enzymes and the substrate, they form a complex called enzyme substrate complex, and then the enzyme converts the substrates into the products. And the substrate or the reactant goes and binds to a special region on the enzyme, and that is called the active site. And when the substrate goes and binds on the active site of the enzyme, there is some change takes place. There is some change takes place in the active site of the enzyme, and that makes the substrate fit better into the active site of the enzyme, and that's called induced fit. So induced fit of a substrate brings chemical groups of the active site into positions that enhance their ability to catalyze the reaction. So this is the this is an enzyme, and this is the place where the substrate is going to go and bind. So this is called the active site. So this is the enzyme and substrate complex. So this, you can see that this is the enzyme, these are the substrates, and this is the active site, and you can see how the substrates are going and binding in the active site. This is the enzyme substrate complex. The active site basically lowers the activation energy. Because how does it lowers the activation energy? This is an important thing to understand, because Otherwise, if there is no enzyme, so you can see the substrates are here, right? They're moving, they're just moving. So for these reactants to react, they need to react, they need to collide with each other, and then they're going to form new products. So because if there is no enzyme, they will keep on moving, and it's a chance factor when they collide with each other and form the products. But if there is an enzyme present, it provides a kind of a platform where these substrates can come and bind and react. And this is the way enzyme functions. This is the way enzyme lowers the activation energy, because enzyme provides a platform where these substrates can come and sit and they can react. So enzyme provides a platform where these reactants or the substrates become accessible to each other, and that's the way the enzyme lowers the activation energy. And on the enzyme, these substrates, they are converted into products, and once the products are formed, they leave the enzyme, and the products, they leave the enzyme, and then the enzyme is again free to catalyze another reaction. So enzyme does not get itself used up in the reaction. Okay. So substrates come, bind on the enzyme on the active site, they react, they form new products, the products they leave, and then enzyme is ready to catalyze the next reaction. So how does the enzyme lowers the activation energy? By orienting the substrates correctly, straining substrate bonds, providing a favorable microenvironment, and covalently bonding to the substrate, as I just explained you. Okay, the rate of enzyme reactions. These some of the things you have already studied in the lab, that the reactions which are catalyzed by enzymes, they can be affected by many factors. They can be affected by substrate concentration, they can be affected by temperature, they can be affected by pH. So basically, if we increase the substrate concentration, the rate of reaction increases initially, but a point comes after which, even if we increase more substrate concentration, the enzymatic reaction does not increase. So the effect of substrate concentration on an enzyme activity is that initially, with the increase in substrate concentration, the rate of reaction increases. But after that point, if we keep on increasing the substrate, at a point will come after which, even if we increase the substrate concentration, the rate of reaction will not increase. The temperature and pH. So every enzyme has an optimal temperature and optimal pH where they function the best. But if we take the enzyme and put it into like a very extreme conditions of temperature or pH, the enzymes do not work, because majority of the enzymes are protein in nature and they get denatured. But there are some enzymes, like majority of the enzymes, they function best at 37 degrees Celsius, which is our body temperature also. But there are some enzymes which have been extracted from thermophilic bacteria. They can function at very, very high temperatures. And majority of the enzymes, they function best at the pH of 7. But there are again some exceptions, like the enzyme called pepsin. It's a stomach enzyme. It can work at acidic pH of 2. Then there is trypsin. It can work at, it is an intestinal enzyme. It can work at a little alkaline pH. Okay, now there are some non-protein helpers of the proteins, non-protein helpers of the enzymes. So they help in the activity of the enzyme, and they're called cofactors and they're called coenzymes. So cofactors, they are non-protein enzyme helpers. They include your metals, like your magnesium, zinc, copper. So some of the enzymes need zinc, they need manganese, they need iron to function. And they are basically non-protein helpers of the enzyme, and they're called cofactors. And then there are some organic non-protein helpers, and they're called coenzymes. So all these vitamins, they are there in the so these vitamins, they have coenzymes. They have the organic molecules in them which can help in the enzyme activity, and they are called coenzymes. So, so here you must think that why do we eat multivitamins and multiminerals? Because multiminerals, manganese, zinc, they are cofactors for the enzymes, and these vitamins, they are coenzymes for the enzymes. They help in the activity of the enzyme. So that is why we eat, we need to take these supplements. Okay, now the enzyme activity is very important, right? So we talked about enzyme activity. Enzymes are very good because they catalyze the reactions. Our cells are nothing but chemical factories because our cells are being driven by all the chemical reactions. Millions of chemical reactions are taking place inside our cells. There are catabolic reactions, there are anabolic reactions going on. So enzymes are good. So, but in some situations, we need to stop the enzyme activity too. Okay. So what can be those scenarios? You need to think about what can be the scenarios where we would like to inhibit the enzyme activity, and how to inhibit the enzyme activity. The enzyme activity can be inhibited by enzyme inhibitors. Okay. Now let's talk about what can be the situation, or what can be the condition where we need to inhibit the enzyme activity. So the very typical example I would like to give you is in the disease called cancer. Now, cancer is a disease which is driven by overexpression of many enzymes. So in that situation, if the overexpression of enzyme is making the cells divide abnormally, then that situation we need to inhibit the enzyme activity. And also you know about the antibiotic called penicillin. Now, what is penicillin? It is again an enzyme inhibitor. It inhibits a very important enzyme in the bacteria which is responsible for the formation of cell wall in the bacteria. So in this situation also we need to inhibit the enzyme activity, because if we inhibit that enzyme which is responsible for making the cell wall in the bacteria, we can kill the bacteria. So in that situation we need to stop the infection. Again, here we need to inhibit the enzyme activity. Now the enzyme inhibitors, they are of two kinds. They're competitive inhibitors and they are non-competitive inhibitors. Now the competitive inhibitors are those inhibitors which go and bind on the active site. So here you can see this is a normal binding. So this is an enzyme. This is an enzyme, and this is the shape of the active site. So this is the active site. This is the enzyme. So now you can see here the substrate goes and binds here. Okay, the substrate goes and binds here, and then the product will be formed. This is normal. Now, what is a competitive inhibition? In the competitive inhibition, the inhibitor goes and binds to the active site, and it goes and sits there. Okay, so because its shape is compatible to the active site, so when the inhibitor goes and binds to the active site, there is no place for the inhibitor to go and bind. The inhibitor has no place. So the inhibitor has actually blocked the active site. Okay. So this is competitive inhibitor. When the inhibitor and the substrate, this is the inhibitor, this is the substrate, when they're competing for the same active site. So when the inhibitor is sitting, no substrate, no reaction. Non-competitive inhibition or non-competitive inhibitors are those inhibitors which do not bind to the active site, but they bind to a site. They bind to a site other than the active site. Okay, you can see here this inhibitor is binding here. Okay, so when this inhibitor binds here, what it is doing is it is changing the shape of the active sites. You can see how the active site is here. Now the shape of the active site is changed, and as a result of that, even if the active site is free, because of the change in the shape of the active site, this substrate cannot go and bind here. This substrate cannot go and bind here, and as a result of that, this substrate cannot go and bind here. As a result of that, no reaction, because substrate is there, active site is free, but they are no more compatible. So it cannot bind here. When it cannot bind here, no products, no reaction. So that's all in this chapter. Please let me know if you have any question.